directions we're going to talk about. But we have the cecum. Here's the appendix hanging off of there. The ascending, transverse, and descending colons named for the direction food moves through that. And we'll talk about this more in just a second. The sigmoid colon, which I always remember because it's kind of S-shaped, at least an S on the side. The rectum, and then the anal canal. So this is my first term doing 233 here, but typically in the other schools I've worked at, they hit these histological layers pretty hard in all the practicals, so, or quizzes. So let's make sure you know the layers. So remember, the innermost layer is called the mucosa. The middle layer is the submucosa. Then you have a layer called the muscularis, or the muscularis externa. In the vast majority of the GI tract, there's two layers, an inner circular layer and outer longitudinal layer, named for the direction the muscle goes around that. So the inner circular, if my arm was the duodenum, is going to go in circles around that. The outer longitudinal is going to run this way. So depending on which plane you cut the intestine in, if you cut it this way versus this way, you're going to be cutting with the grain in one and against the grain in the other, or through, right? So it'll be easy to see histologically the differences. In between there, you have a nerve plexus called the myenteric plexus. It's on your list. Remember, the mucosa gets broken up into three sublayers. So there's the actual epithelial layer here, sometimes called the mucous membrane. There's the lamina propria here. And then there is the muscularis mucosa, really, really thin layer of smooth muscle. Allows you to make very small local changes in the anatomy of this, or movement of things through here. I think the most helpful thing the muscularis mucosa clearly does is it's a pretty clear line that separates the mucosa from the submucosa. Also in the submucosa, you're going to see tons of blood vessels. Why are you going to have lots of blood vessels there?
That's its job. Yep.
Yeah, to absorb the food, right? So you've sucked food in through these cells, you got to get it into the bloodstream. Okay, so superior and inferior labia, just the lips. Labial frenulum, they're the little, the tissue that connects the lip to the gingiva or gum. Gingiva is, again, the gum. Raphe is just the seam right here that connects the two parts of the hard palate. The hard palate we already talked about last class. It's just a bony separation between the nasal and oral cavity. Soft palate is behind that. You can feel where the bone ends here. And then the uvula is the end of the soft palate here. Let's see. There's two arches in here. So there is the palatoglossal arch here, and then the pharyngeal arch behind that, which you can kind of see. I don't know if it's pointed out on here. Pharyngeal arch, yes, back here. So those two arches basically create a little cavity or cave that the palatine tonsils are located in. And you can remember these are the palatine tonsils because they're right by the palate, right? Versus the pharyngeal tonsils and the lingual tonsil that we talked about last class.
Are those usually the ones that are removed when people talk about having the tonsils?
Typically those are removed, and then if people say their adenoids are removed, those are the pharyngeal tonsils.
Okay.
So sometimes they take both of them out. Just for a later point of reference, the submandibular salivary glands empty underneath the tongue there. Or empty.
Is it actually on the frenulum?
What?
Is it actually on the frenulum?
Oh,
or
it's
is it
kind of at the base of the frenulum. So the histology of this, all the upper structures, which the only slide we actually have is for the esophagus, but everything basically above the diaphragm is going to have stratified squamous epithelium lining it. So that's going to be a really big clue, right? So you're looking at the esophagus here, here's the lumen, here's the esophagus. We used to at PSU, and I don't think they have them here, we had these great esophageal cardiac stomach kind of transitions where you just see, like, you see the lower esophageal sphincter, which is kind of a big lump, and you see stratified squamous on one side and simple columnar on the other, and it was a really abrupt transition, so it was kind of a neat thing to look at. We don't have those slides here, but non-keratinized stratified squamous. Why is it not keratinized?
Because it's not on the outside.
Right, not on the outside. You're not gonna lose fluid through there or dehydrate through there. Here's a close-up of that. Again, you see the stratified squamous. Here's the lamina propria, the submucosa, and then the muscularis externa on the outside. Same thing, just a close-up of the same picture. So stratified squamous, and then you can see that to the bottom of the mucosa here, right? The lamina propria, and then the muscularis mucosa. In the rest of the GI tract, really the muscularis mucosa is a little easier to see than this. It's a little more distinctive a line. All right, let's talk about the stomach. So, the stomach, I don't remember if this is on your list. There's a sphincter right there, either called the lower esophageal sphincter or the cardiac sphincter. Might not be on your list. The cardia is the region right where the food first enters. If you kind of imagine that the stomach is a J shape, the top part of the J up there is going to be the fundus. The straight part is the body, and then the hooked part is the pylorus. There are lots and lots of folds in the stomach. They're called rugae. We're going to see that word rugae in several different sections. Rugae, again, is a general word just meaning fold. Sometimes they're called gastric folds in some textbooks. You can see the pyloric sphincter at the end here. The greater and lesser curvature, the lesser curvature is a smaller curve here. Greater curvature is the under curve here. And then in the stomach, you can see there are actually 3 muscle layers. So there's an inner oblique layer, and then it just pushes the other 2 out a layer. So the circular is now the middle. Remember, in the rest of it, the circular layer is the inner layer. And then the outer longitudinal. The reason the stomach, or any theories as to why the stomach has more smooth muscle than the intestines?
It does a lot of.
Yeah, it grinds stuff down. It actually does physical digestion. Once the food makes it into the intestines, it's all chemical digestion, right? So you're chemically breaking apart bonds.
There's on our list it says gastroesophageal junction. What's that?
Gastroesophageal junction is where it's just talking. So where the esophagus and the stomach meet, right here.
So is that there's also gastroesophageal sphincter? Those are the same things, basically.
The sphincter is the smooth muscle there, and the junction is just that whole area. Yeah, and like I said, I don't think we'll have to look through the slide boxes. I don't think we have a slide that shows that. It's a neat thing because it's so abrupt. It's easy to see compared to some slides for some sections that aren't quite as clear. I wanted to show you quickly the greater and lesser omenta. So it's kind of a fatty layer. So the greater omentum attaches to the greater curvature of the stomach. The lesser omentum attaches to the lesser omentum. Lesser curvature. So even if you feel like your abs are nice and tight, you might have a nice little sheet of fat underneath there. If you guys get the chance to do cadaver work, you'll never ever forget these because these are always in your way. You're always trying to move them.
What is their function?
Fat storage and protection. Okay. This is an image I have from my lectures at Clark, so I'm glad it's in here because this kind of gives you, I don't know if your lab book has something similar. This shows you kind of a cartoony version of the histology in the stomach. So you have gastric pits, which are fairly large indentations into the surface of the stomach, and beneath those you have these long glands. Most of the time, you're not going to see the gland going down straight because they kind of run all over the place, so you'll see circles beneath that. Sometimes you get lucky and you actually see them cut, where you can actually follow one down, but usually you're going to see just a bunch of circles underneath here. There are lots of different cells you need to identify today in the stomach. So chief cells down here, they are shown in purple, although that one's a little misleading. They're shown in purple. Here, typically they're in the bottom half. The only thing that's a little misleading about this, typically they're concentrated more in the bottom half of the gastric glands. Anyone remember what these guys secrete from lecture? Chief cells?
Hydrochloric acid.
Pepsinogen, yeah. So pepsinogen, actually the inactive form of pepsin, right? typically in the next section up, so the kind of middle third, you have parietal cells. Parietal cells typically stain reddish, and chief cells typically stain bluish purplish. Parietal cells secrete HCl, and the reason I remember that is because this is sort of a goofy mnemonic, but parietal cell starts with a P, hydrochloric acid starts with an H, pH, you know, and hydrochloric acid lowers the pH. I don't know, it sticks with me then. If I used to always get them confused before then. You also have things called enteroendocrine cells. So those are specialized endocrine cells in the GI tract. Typically, you're going to see these called G cells because in the stomach they secrete, for the most part, gastrin, which is the hormone. So that's what makes them the enteroendocrine cell. Anyone remember what gastrin does? Gastrin? You guys talk about that in lecture? So gastrin causes pepsin to be secreted. It also weakly, pepsin and hydrochloric acid to be secreted. It also weakly stimulates the gallbladder contractions and pancreatic enzymes. So it's kind of kickstart the rest of the digestive enzymes and processes. Sometimes they do tests on the stomach gastrin if people have like a peptic ulcer or they suspect that they have peptic ulcer disease. They'll look at stomach gastrin levels or gastrin levels in the bloodstream to see if you're over secreting that.
And you said that's the G cell, right?
The G cell.
So they're usually
Same
at
as
the bottom of the
enteroendocrine cells.
bottom of the gland.
Yep, very bottom of the glands. What was I going to tell you about these? These tend to be, I'm probably unlikely to ask these. I'll look in your slides and make a final decision later. I'm probably unlikely to ask these. I haven't really ever seen a huge distinction between the G cells and the chief cells. So I'm more likely to ask you chief or parietal cells. The last cell type that you're going to identify are called mucous neck cells. And basically it's all the stuff shown in pink here. And even on the surface they secrete mucus too. So the point of those cells is to secrete a thick layer of mucus so the digestive enzymes and hydrochloric acid don't eat away at the stomach lining. So any of these are mucous neck cells. Once they're on the surface, they're called mucous surface cells, which isn't on your list, but the cells look exactly the same. They're doing the same job. All right. Oh, you know another thing that can make gastrin levels in your blood go up? Any ideas? Chronic use of antacids. So you use tons and tons, like, you know, people who take Prilosec for years, you know, that's a stomach acid reducer. Your body really pumps up the amount of gastrin because it's saying, oh, we're underproducing hydrochloric acid, so then you produce a lot more gastrin, which is one reason when people come off those drugs, they have a big acid rebound, right? They have tons of kind of over-secretion of acid. Okay, so let's just take a look at this histologically. This is low magnification view, right? So here's the whole mucosa here. Here's the submucosa. Oops, submucosa is kind of thin. And then you have three different layers. So inner oblique, middle circular, outer longitudinal. The outer layer here is a serosa. So what's the difference between a serosa and an adventitia? You guys probably talked about this in lecture. Yeah. So serosa secretes serous fluid, right? And they're basically in the abdominopelvic region. All of those organs, the outside of them are lined with a serosa. Everything above the diaphragm, basically, that we're going to look at, so the esophagus, has an adventitia on the outside. So did the trachea, remember? Yes.
So that's the only difference, is that the cirrhosis secretes serous fluid and
Yeah, so you can, if you're really good at your histology, you can see some histological differences because of that. You guys will recognize just basically all your GI organs other than the esophagus, or the outer layer is the serosa. And then the functional difference is these guys secrete serous fluid, and the other one is more just for protection. And the serous fluid is going to reduce friction, right? So when all those damp organs are kind of moving across each other. Okay, here's another low magnification view. I like this because you can really see the submucosa. The other thing I like about this is it gives you the idea that you're not always going to get a perfectly straight section, right? So you have big folds in your stomach, remember, called rugae. So this whole structure is a ruga here. You can also see your gastric pits indented there, and then all these structures here that aren't quite magnified enough are gastric glands beneath it. And again, you're basically seeing really small Cheerios down there. You can see the muscularis mucosa here, even on this low magnification view. You can see the submucosa, and then our muscularis externa here on the outside, almost the outside, and the serosa is the very outer little bit there.
And is there 3 layers in that?
There are
muscularis?
3 layers
Do you
in
see?
there.
Can you identify
Yep.
them?
Inner oblique, middle circular, outer longitudinal. All right, let's take a closer look at this so we can take a look at the different cells you guys have to identify in here. So again, all of your, everything from your stomach down to your rectum are going to be lined with simple columnar epithelium. So these are the gastric pits here, right, and the gastric glands beneath them. The chief cells are staining, in this picture, nice dark purple. You see a lot more of them in the bottom third here. Parietal cells are pinker staining, a little hard to see on this one. And then you have those mucous neck cells that you can easily identify up here. And those are those nice, tall columnar cells. I have one more slide that this is a little closer. Same slide, right? Slightly different spot. But anyway, so you can see all those nice darker staining chief cells. We can see our parietal cells, which again, they tend to stain pinker. That's not coming through on this overhead as well as it does on the computer. We'll look at that histologically, or you guys will see that in the scopes. And then those mucous neck cells should be pretty easy to identify because they're simple columnar, and they're going to be towards the very top. All right, so moving on to the small intestine, I wanted to point out a couple of things. There are 3 different scales at which the surface area has been increased, right? So the whole point is you want a ton of surface area so that you have a lot of space for absorption to occur across. And where does the bulk of absorption occur? Small intestine, right? So very little occurs in the stomach, and the bulk of that actually occurs in the jejunum, right? Which if you look in the jejunum, which for some reason isn't on our list histologically, just the duodenum and ileum are, but they have the longest, tallest villi. Anyway, there are certain things that get absorbed in the duodenum. You could talk about those substances in relation to gastric bypass. Interesting thing to think about because they usually bypass the duodenum as well. Anyway, that's an aside I'd talk to you about if you wanted to later. But anyway, so you have these big folds that basically cause the chyme, which is the chewed up food with the enzymes and all that, to kind of spiral around to basically get it in contact with as much of the surface area of the intestine as possible. So these big folds are called plicae circulares. Some other texts call them circular folds. They're plicae circulares on your list. So those plicae circulares, so this whole thing is a big plica circulara here. Right, so you have all these little finger-like projections coming off of those going into the lumen, right? So that's the next scale at which you're increasing surface area. And then each individual cell, so this is one of those tiny, so what we're going to do is we're going to take one of these tiny villi, so like this one right here, and we're going to look at it really high magnification now. So each villus is lined with cells that have tiny little microvilli on them. So those 3 scales, so that's a ton of surface area. I forget the exact number. I think I looked in one book that said something like 2 million cubic centimeters of surface area, an amazing amount of surface area. You can't, these are on your list, the microvilli. You can't really see these. Some people claim, they call this a brush border as well. They can, it looks a little fuzzier at the surface. I don't think we can really see that on our slides. You should know that the microvilli are there, though. All right, let's look at this histologically. So you have to distinguish between the duodenum and the ileum. So a few things that are going to help you. So you're going to look at the shape of the villi. So the villi are the finger-like projections into the lumen. Do you think the villi are always going to look perfectly finger-shaped and straight? No. Depends on how they're laying when you cut it and how perfectly they cut it through. Okay, so basically the further down the GI tract you get, the more goblet cells you're going to have. Why do you think you need more goblet cells the further down you get? More mucus. The more fluid you pull out of there, the more mucus you're going to need to lubricate that chyme through there. Also, the further down you get, the more lymphoid tissue you're going to find. In the ileum, we have you identify Paris patches, very obvious big lymphoid nodules. Why do you think you need more lymph tissue the further down you get?
More bacteria growing in it.
Yeah, you've eaten food and kept it at 98.6 degrees. So the longer it's in there, the more the bacteria get to go crazy. So the more contingent of your immune system you need there. Also, the shape and size of the villi. So the villi of the duodenum are going to be longer. In the ileum, they're a little less regular. They're a little squatter. Another thing that you only see in the duodenum are these glands called Brunner's glands that I don't think are on your list. You guys have Brunner's or duodenal glands?
Duodenal.
Okay, same thing. Anyway, these glands, do you remember from lecture what they do? So you only have them in the duodenum, and it's because they secrete an alkaline substance that helps decrease the pH of the acidic chyme coming out of the stomach. So the acidity of the chyme, by the time it gets all mixed with everything in the stomach, is about pH 4, right? Your stomach pH is about 2.5, but then the food is going to raise that some. But it's about pH, 4-ish. So these Brunner's glands, along with sodium bicarb that's secreted from the pancreas, all help to raise the pH up to about 8 at this point. 7.5 to 8, depending on the book you look at. Yeah, so of course you're not going to need these by the time you get to the jejunum and ileum because the pH is already raised. So you'll see those in the submucosa of the duodenum. The glands we saw in the stomach, what layer were those in? Those were in the mucosa. Yeah. So those are the only glands we have today that are actually in the mucosa. These are in the submucosa. So this right here is the mucosa. This is the submucosa. And then you should be able to see two pretty distinct layers of smooth muscle here, the inner circular and outer longitudinal layers. And then the very outside here is the serosa. Here's another one. Same thing, right? Oh, I forgot to mention the crypts of Lieberkühn or intestinal crypts. So they're just at the base of the villi. So here's the villus, here's the crypt. The crypts go down. The crypt, one of the functions of the crypts is you have highly mitotically active cells down here that are pushing cells up onto the intestinal villi. Because obviously these cells are under kind of constant assault, right? So there's a high turnover rate of those. One reason people have a lot of GI complications when they take chemotherapy is these are some of the faster dividing cells in your body, right? And chemotherapy is going to disproportionately affect anything that divides quickly. Just like hair, that's why your hair falls out.
Okay, Amy?
Yeah.
There's two things in the small intestine that I couldn't find. There's the major duodenal papilla and the hepatopancreatic sphincter.
Yeah, we'll talk about those in a
They're coming
sec.
up? Okay.
Yeah. We'll talk about those when we talk about the liver and gallbladder. Okay, here's just a close-up of that. You can see not a ton, so this is still the duodenum, not a ton of goblet cells. I wanted to show you guys one picture of a lacteal just quickly. So a lacteal is a lymphatic duct that runs up the middle of the villi. And here you can see it pretty clearly here. Ours, if we're pointing to something in the middle of a villus, is probably what we're pointing to. You remember what those lacteals do from lecture? They're basically they're a lymphatic vessel for one, but they're also a specialized way that fat gets absorbed through the villi. Did you guys finish your digestive lecture already?
We haven't done it yet.
Oh, you haven't done it yet. Okay. What did you do last class or yesterday?
We did a lot of the enzymes and stuff, not so much anymore.
Oh, okay. All right. Well, you'll have a head start on this. So here's a pretty low magnification view of the ileum. And again, you can see these villi are less regular. They're going to be a little shorter and squatter. You also have these very obvious Peyer's patches, which are lymphatic tissue. Oh, I suppose I can put it on the slideshow version. The other thing you can see in this one, so you can see the close-up of the Payer's patch. One thing you might be able to get a sense for, although the resolution is kind of low on here, you might be able to see that there are lots more goblet cells, those lighter staining cells. You guys will notice that for sure when you're looking at those. Okay, let's talk about the large intestine quickly. So the appendix, also called the vermiform appendix. Anyone know what vermiform means?
Worm-shaped.
Yeah, worm-shaped. Worm-shaped appendix here. Anyway, the appearance of their appendix really varies greatly. We had a cadaver, I don't know, 2 terms ago at Clark that had this appendix that was like this long. It's crazy.
Wow.
Still had it, but anyway. So here's the ileum, here's the cecum. Right where they join together is an area called the ileocecal valve or sphincter. Is that on your guys' list? Yeah. That's actually pretty obvious, and you can see it well on the models. I don't think we have slides that show that. It's pretty obvious in the slides, too. Cecum is this first kind of pouch portion. Then you have the ascending, transverse, and descending colons. Just you can remember that they're named for how food moves through them. You have the right or hepatic colic flexure and the left or splenic colic flexure. Obviously, right and left, those are the sides they're on, and then the liver's on this side, the spleen is on this side. That's why they have those names. Anyway, the sigmoid colon, if you use your imagination, I always remember this because it looks like an S laying on its side. This is just portion here. You have the rectum and then the anal canal. I think that's... Oh, interestingly, the outer longitudinal layer of muscle is reduced just to 3 bands here, which are called the tenui coli. So you have 3 bands of the outer longitudinal muscle layer there. And they're shorter than the intestine, so they kind of make these pouches called haustra. So those pouches called haustra, I always think haustra sounds like it should be something at IKEA. But anyway, the other thing you see here are epiploic appendages, which are just the little fatty sacs that are actually, they're fat-filled pouches of the visceral peritoneum that hang off of the large intestine. Let's look at this histologically. So one thing you'll notice right away is there aren't any villi. So you still have intestinal crypts or crypts of Lieberkuhn, but you don't have villi. So it looks a lot more regularly arranged. Why do you think you don't need to have villi at this point?
Absorbing.
Yeah, you're doing way less absorption, right? You absorb water at this point and some vitamins, but that's about it. And you're going to see tons and tons and tons of goblet cells. Sometimes you find a section where it looks like there's almost more goblet cells. Oh, let me put one in there. Huh. I have 2 in my slide. Anyway, okay. So deciduous teeth, you guys have quite a bit as far as teeth goes, but we don't have any teeth histology, which is a shame. They're kind of pretty to look at, and they're really distinctive looking. Deciduous teeth, so you guys do need to know the dental formula. It lists kind of the parameters for that in there, but it's 2102. So it's always the number of incisors, canines, premolars, and molars. So before you get your adult teeth, you actually don't have any premolars. So you have, and it's in each quadrant of the mouth. So, and it's usually top over bottom. So 2 incisors, 1 canine or cuspid, 0 bicuspids or premolars, and then 2 molars. Who here already works with teeth? Usually there's a few people. There you go. This is your bread and butter right here. All right.
So deciduous.
Deciduous just means the teeth that you lose. Hey, there are people that actually lose 2 full sets of teeth. I had a person in my class a year and a half ago who her whole family had 2 full sets of deciduous teeth. She said so their family would lose their deciduous teeth, their first set a little earlier than average, and then the other one, she said it was kind of embarrassing, like in junior high, to still be losing teeth. But anyway, permanent teeth. 2123 over 2123 is the formula. So it's 2 incisors, 1 canine or cuspid, 2 premolars, and 3 molars. The 3rd molar is your wisdom tooth, right? How many people still have their wisdom teeth, just out of curiosity? I do. So most people don't. Oh. You guys might be surprised to hear this, the dentist said I have a very big mouth, so. I know, surprising. Okay. We, I don't want to take any longer. The picture of what is on the tooth, the anatomy of the tooth is pretty easy, very straightforward. Ask if you have questions, but I want to make sure the main point of this, I mean, I wanted to familiarize yourself with this, but I also really wanted to make sure the histology was in here because your book doesn't have all the histology. Salivary glands, parotid, submandibular, and sublingual glands. Par means next to, right? Anyone know what ot means when you hear that in something?
Ear.
Ear, right, so next to the ear. This gets swollen if people have mumps. So in old movies where you see people with mumps. Or John McCain had a calcified parotid salivary gland. Anyone notice he had a weird cheek? Yeah, that was calcified parotid salivary gland. Submandibular salivary gland, remember I told you where those ducts empty underneath your tongue? Not the sublingual, you have many small ducts, but submandibular, you have those two big ducts underneath your tongue. So people who can gleek, it's a submandibular gland they're using there. I have 5 brothers, so I know all about that. But, so, all right. I was really surprised when I didn't get a chance to look through the slides till today. I think the only slides we have are parotid salivary glands. The other places I've worked, we've had you distinguish between the 3 sets of glands. I'll double check that, but I think we only have parotid glands. So the different glands, and you probably need to know this for lecture, secrete differing amounts of either mucus or salivary amylase. So the mucus cells are these kind of white marshmallowy cells, and the cells that secrete the salivary amylase are these serous cells. So they're dark staining cells. I always remember serous and serious. It's dark and brooding, so I always remember those ones are dark. Anyway, when they're in this kind of half moon shape on top of a kind of circle of serous cells, they call it a serous demilune. That just means half moon or part moon. So it kind of looks like a little crescent moon sitting on top of there. It's going to be by far the easiest. Let me see here. So parotid glands, they make about 25% of your saliva, and they make what do I want to say here? I think they make mainly serous salivary amylase, so they secrete a lot of the submandibular glands secrete about 2/3 of your saliva, and they secrete a mix of mucus and salivary amylase. And then the sublingual glands, a little less than 10% of your saliva, and it's mainly mucusy that kind of coats the bolus of food in kind of a mucusy package before you swallow it. All right. So I put all 3 sets of glands in here. Here's another one that you can really see the serous demilunes easily, right? You can see the difference between the mucus cells and the serous cells really easily. And there's a serous demilune, the little crescent moon there. Because I don't think we have all 3, we'll kind of speak to this next one here. Parotid has, you secrete very little mucus from the parotid gland, so there's less mucus cells in there. Mainly serous cells. It doesn't mean you can't find some mucus cells, right? These lighter staining ones. It's also not uncommon to find fat in these, so don't confuse fat with mucus cells, right? You guys are familiar already with what adipose looks like from your earlier histology work. Oh, by the way, the ducts. What kind of epithelial tissue is a duct going to be lined with typically? Simple cuboidal. If you find, oops, that's a blood vessel. If you find a really, really large duct, sometimes you can find stratified cuboidal. Kind of fun to see. It's a pretty rare type, but if you find a really big salivary gland, sometimes you can find stratified cuboidal. All right, let's talk about the liver and gallbladder. So 4 lobes of your liver, right, left lobes are the biggest. The right is by far the largest. You have the quadrate lobe and the caudate lobe. Quadrate means square, right? Caudate means towards the tail, so this is posterior. You also have the gallbladder embedded in the right side there. One of the many, many jobs of the liver, and the liver I think is really, such an interesting organ. Does so many things we could talk about later. But anyway, so one of the many jobs is to secrete bile. So you have bile secreted into small ducts that lead into bigger and bigger ducts that are going to empty then into the right and left hepatic duct. Those are going to join together to form the common hepatic duct. Anytime you see hep in something, it means liver, right? Here's the gallbladder, and then you have the cystic duct right here. connecting that. Where the cystic duct and common hepatic duct come together, from here to here, is called the common bile duct. So right where the common bile duct is coming through the wall of the duodenum, you have something called the hepatopancreatic ampulla or ampulla of Vater, right here. So what's an ampulla? Anyone know? Anyone? It's a flask-shaped, oh, nice. It's a flask-shaped opening. So if you look at the better, there's a few of the better models. Oh, by the way, the pancreatic duct joins together with that too. So we have this flask-shaped opening called the ampulla of Vater or hepatopancreatic ampulla, which, although that's a mouthful, right, it's bringing stuff from the liver and from the pancreas, so it's not, I mean, the name makes sense. What else is on there? Oh, major duodenal papilla is just a bump in the duodenum where that comes in. So papilla is a general word for a bump. We'll see that word again and again. And then the sphincter of Oddi, or the hepatopancreatic sphincter, is just a tiny little circle of muscle that opens and closes to let bile and pancreatic enzymes in or not. By the way, there's also an accessory pancreatic duct here called the duct of Santorini. And it's so that you can close this up, not put bile into the chyme, but still allow the pancreatic enzymes in. So you can close this sphincter and leave this one open if you just want pancreatic enzymes but no bile. All right. Let's look at the histology of that, and then we are just about done. So the functional unit of the liver is something called a liver lobule. It's roughly hexagon shaped, so roughly 6 sides. In the middle of it, you have something called a central vein that is basically a branch off of the hepatic portal vein. And what does the hepatic portal vein do? Yeah, it brings blood from all the GI organs and spleen, for that matter, through the liver, right? For all the magic the liver does. So to take all amino acids out of it, and to take glucose out of it, to detoxify, etc., to break down worn out red blood cells out of it. This from here, this is going to be filtered through all these hepatocytes, right? And then it's going to empty into a branch of the portal vein here. Oh, sorry. Let me think. Yeah, those go out, these go in. Sorry. We have a portal vein here and the central vein. So the blood is running this direction. So here's a branch of the portal vein here at each of the corners of the sinusoids. It's going to run towards the central vein. From the central vein, then it's going to lead into larger and larger blood vessels until it leads into the hepatic vein to get dumped back into the vena cava. My flow direction was opposite there. Yeah, so portal vein here, a branch of the hepatic portal vein. The blood is going to filter through these openings called sinusoids so the hepatocytes can do their magic. Also, hepatocytes are producing bile, and they're shipping it out through tiny little things called bile canaliculi, the opposite direction, and they're dumping it into small ducts in each of those corners of the lobules. There's also a hepatic branch of the hepatic artery there, because these cells obviously need a lot of oxygen because they're very metabolically active. So this group at each of the corners of the lobule is called a hepatic, a portal triad. So you have hepatic portal, a branch of the hepatic portal vein, hepatic artery, and you have a duct. And let's look at that. That's what you're going to be looking at histologically. So you guys can, in this one, you can see the lobules pretty clearly, right? Lobule. In the middle here, you have a central vein, and then at each of these corners, you should be able to see the portal triad. You can see them better on some than others, obviously, like any histology. Let's do a close-up of one of those so you can see them. Oh, and you can even see the sinusoids in this picture, those little white spaces, right, that the blood is going to be flowing through. So central vein here, portal triads here. We'll look at a way closer up of that. What do you think the bile duct is going to be lined with? What kind of epithelial tissue? Simple cuboidal. Right, so that one's going to be easy in the triad, the simple cuboidal epithelium. The portal vein is going to be bigger than the arteriole, so you're going to notice that. So here's our little bile duct, right? We see the simple cuboidal epithelium, the little hepatic arteriole, very small little artery. You can even see the more smooth muscle in the lumen here, or sorry, around the lumen. And then here's our hepatic portal venule. So branch off the hepatic portal venule. Doesn't have to be this much bigger. That one was kind of cut sideways, but still it's way bigger than either of the other two. Another close-up of that, right? So you see the bile duct, the portal venule, and the hepatic arteriole. Pretty easy to distinguish. Remember, a lot of times arteries, post-mortem, that smooth muscle contracts and pushes the nuclei into the lumen. Not always, but oftentimes that happens. So don't confuse that with simple cuboidal, right? Where you have a nice round nucleus in the middle of the cell. Sometimes people see this and just, because it's not flat, want to call it cuboidal. It's simple squamous still. The nuclei just get shoved up when that smooth muscle contracts. All right. We have our central vein there, and the sinusoids are just the spaces. Remember, hepatocytes are the cells that are doing kind of the general job of the liver, and then there are specialized cells in there, hard to see here, called, let me see if they're actually on the list.
Is it Putnam? Cooper?
Yeah, Kupffer cells. Yeah, the Kupffer cells, you're going to see those in the lumen. This doesn't show it well. Here's of the sinusoid, so there's one there. Here's one here. Your slides, you'll be able to see them better than this. These, if you're looking at an older textbook, you might hear them called, which I know is a long name, but stellate reticuloendothelial cells. So don't panic if there's something else. I mean, if they're called something else. But they phagocytize debris from the blood. So they're right there kind of hanging out where the blood flows over them. All right, last but not least, we have the pancreas, which should be really, really easy, right? You guys have seen the pancreas before. I'm not going to go over the general anatomy, really easy, head, neck, body, tail, etc. Remember, you have the lighter staining pancreatic islets, and you have ducts in there that, like all the ducts we've talked about, are going to be lined with simple cuboidal. So these are all pancreatic islets. What do the islets do again? They make the endocrine secretions, right? Insulin and glucagon are the biggest of those. The rest, 99% of the pancreas secretes digestive enzymes and sodium bicarb that gets dumped into these ducts and eventually into the main pancreatic duct or accessory, and then in most cases through that hepatopancreatic ampulla, right, that we talked about. All right, so this is all online. I know this is long. I don't know how to make it shorter. Sorry, there's close up of that.